Welcome, dear students. Good morning. So we were talking about HPLC, high performance liquid chromatography. And in yesterday's lecture, we have seen some of the important techniques. Okay. Can you tell me which techniques we have seen yesterday in HPLC?
Ion exchange chromatography.
Ion exchange chromatography. Very good. Anything else?
Gel filtration.
Yes, gel filtration chromatography. So today we are going to see another important technique, which is especially useful for certain proteins which are more hydrophobic in nature. Okay, and this is especially considered to be a high resolution method because different proteins have different hydrophobic amino acids. Okay, and there is a certain proportion of amino acids which are hydrophobic, and every protein will not have the same number of hydrophobic amino acids. Okay, and on that basis, it is possible to differentiate between the different proteins. That is to say, we can separate different proteins based upon their hydrophobic nature. Okay. Let me go to the next slide. Okay, can you all see that?
Yes, sir.
Okay. So this is a high resolution method. We have special columns that are used in hydrophobic interaction chromatography, and that is phenyl sapphirose column. Okay, phenyl groups are present on this column. That is also called sometimes as a superrose column. So these names, superrose or sapphirose, these names are actually the brand names of the companies. Okay, so we are of course not very much interested in that at present, I would say. But what is important is the phenyl groups are present on these stationary phases, and these phenyl groups have a property of binding the hydrophobic proteins or hydrophobic groups which are present on the proteins. Okay, so this is a you can say a kind of a specific interaction where the phenyl groups on the column stationary phase will bind to the hydrophobic amino acids or hydrophobic groups which are present on the columns on the protein, sorry. Okay, and what we essentially do is a small kind of manipulation we have to do. That is, if we have the proteins in a high salt solution, okay, the sample protein is taken in a high salt solution. So because of that, what happens is the hydrophobic patches which are there in the protein, sometimes the hydrophobic groups are submerged inside the protein molecule. A protein molecule is a globular molecule, isn't it? You can easily imagine that. Now, inside this globular structure, some of the hydrophobic groups are hidden inside. Okay, because normally the protein is supposed to work in an aqueous environment, isn't it? And therefore, the hydrophobic groups are hidden inside the protein. Now, when we take the proteins in a high salt solution, these hydrophobic groups or hydrophobic patches, they get exposed outside. Is that clear to you? And once the hydrophobic groups are exposed on the surface, then if we put this sample on a column, hydrophobic interaction chromatography column, then the phenyl groups on the column will bind to these exposed hydrophobic groups. Is that clear to everybody?
Yes, sir.
Okay. So now what happens is you can imagine that different proteins are there in the mixture. They have different hydrophobicities or different hydrophobic character they have. And because of that, the proteins are binding to the column. Okay, now you can imagine that there are maybe say 4 to 5 different proteins which are already binding to the column. So how are we going to elute the proteins out of the column one by one? Okay, we want the proteins to come out of the column, isn't it? So we are going to put the mobile phase to the column, and the mobile phase will drive the proteins which are binding to the column outside. But now they are sticking to the column because of the hydrophobic interaction. Hydrophobic interaction is there. So how can we remove the proteins? Anybody would like to answer that?
By decreasing the salt concentration.
येस, कोणी सांगितलं हे? छान उत्तर दिलं. Who was that?
So
Vaidehi. Yes, you are right, Vaidehi. Okay, so I have told you that when we put the protein sample in a high salt solution, then the hydrophobic groups are exposed. So now what we want to do is we want to do the exact reverse of this. We want the hydrophobic groups to be unexposed, or we want to hide the hydrophobic groups so that the proteins will come out of the column. And that we can do by decreasing the salt concentration. Now, what interesting thing we can do is we can slowly decrease the concentration of the salt in the mobile phase, not at once. Okay. For example, I can as well put distilled water as a mobile phase. And if I put distilled water, then probably all the proteins will come out of the column because all the hydrophobic groups will be hidden in the proteins, in all the proteins. But I don't want all the proteins to come out at once, isn't it? And therefore, what I do is slowly I will decrease the salt concentration step by step. Okay, or in a continuous way. Okay, so as the mobile phase enters the column with a reduced salt concentration, the protein which is least hydrophobic will probably come out first. And then as I go on increasing the salt concentration, the proteins which have more and more hydrophobic groups, they will come out one by one in the increasing order of their hydrophobic nature. And if I have a very low salt concentration solution, then probably the most hydrophobic protein will come out at the end when the concentration of the salt is very less. And then if something is still binding, if some protein is there which is highly hydrophobic, it is still binding to the column, then maybe I can put distilled water that is zero salt concentration, and that protein also will come out. Is that clear to everybody?
yes, sir.
Okay, what about the boys? Akash,
Yes, sir. Yes, sir. Clear, sir.
सतीश, रजनीश, अजून कोण आहे? आज भीमभूषण नाही
Yes,
आला. भीमभूषण नाही दिसत आहे आज. ओके, अजून काय अटेंडन्स आपली कमी कमी होत चालली. तेजस, कॅन यू टेक द अटेंडन्स टुडे? तेजस, कॅन यू हिअर मी तेजस?
Yes, sir.
ओके, तू का बरं दांड्या मारत होतीस इतके दिवस?
नहीं सर, कल नहीं थी सर.
हाँ, उसके पहले भी शायद नहीं थी.
नहीं सर, प्रेजेंट थी मैं.
ओके ओके, ठीक है. तो तुम्हारा तुम
कल गलती से रह गई सर.
ओके, सो यू टेक द अटेंडेंस टुडे. ओके.
Okay, sir. Yes, sir.
So then there is one more kind of mobile phase, that is propanol. And if we use propanol as the mobile phase, then the most hydrophobic proteins also will come out of the column. Tejas, can you switch off your mic?
Yes, sir.
Okay. थोड़ा मी स्लाइड्स च्या मोड मध्ये जातोय. Okay, so now what I have told you is this. Just a minute. That in ion exchange chromatography or hydrophobic interaction chromatography, what we do is either we change the pH, okay, pH of the mobile phase, or we change the salt concentration of the mobile phase, and then by altering these conditions, we can elute the proteins. Okay. Now, sometimes what happens is when the protein components, the different components in that mixture, they come out in the mobile phase, that mobile phase already has some salt, isn't it? It may be a high concentration of salt, or maybe it has a low concentration of salt, or whatever it is. Okay. Now the question is, if it is a high salt concentration, or maybe if the pH is too acidic or too alkaline for a certain protein, then is it possible that that will damage the protein or denature the protein? Because if we keep that protein for a long time under those conditions, maybe the protein structure is going to be altered, isn't it? Yes or no?
Yes, sir.
So then the question is, what should we do about it? How can we remove that salt or other ions, or maybe how can we bring the protein in a proper pH? How can we alter that condition, or how can we separate the protein from the salts? That is the question. सगळ्यांना प्रश्न कळलाय का? Have you all understood what I am trying to ask you? Satish? Saida? Saida, have you understood what is the question?
Yes, sir.
So how can we separate that protein, or how can we remove the salts? Or
Yes,
any-
I'm trying to figure out.
Yes.
I'm thinking.
Have you heard about this word dialysis?
Yes, sir.
Yes, sir.
So what we do is we carry out dialysis. Okay. What we can do is we can take the protein sample or the protein component which has come out of the column, and we put it in the dialysis bag. Inside the dialysis bag, what will happen? Outside the dialysis bag, we have distilled water. Inside is the sample. Okay. And then the salt will move from high concentration to low concentration. Okay. So the salt ions, whatever are there, or whichever ions are there in that sample component, okay, they will come out of the dialysis tubing or dialysis bag into the distilled water. Okay. So we have to give changes, a number of changes. That is to say, we remove the dialysis bag from that after some time, because it is an equilibrated process, isn't it? After the equilibrium is reached, no more of the salt will come out. And therefore, you remove that dialysis bag and keep it in another fresh distilled water. Okay, so when you put it in the fresh distilled water, whatever salts are there inside, they will also come out from that bag. Okay, so we have to continue this for some time. Repeatedly, we have to keep that dialysis bag in fresh distilled water, so that whatever ions are there inside, they will come out of that bag or come out from that sample component. Is that clear to everybody?
Yes, sir.
Okay.
Yes, sir.
Okay, so I go to the next method, and that is called as affinity chromatography. Have you heard about this word? Affinity chromatography.
Yes, sir.
When was it in your BSc? BSc or what?
Yes, sir. I've heard it in chemistry.
Okay, chemistry. Yeah, that is right. Okay, so this is again a very, very interesting type of a chromatographic technique where we do not depend upon the different physico-chemical properties of the sample molecule, like charge distribution or surface charges, hydrophobicity, size of the molecule, and so on. But here what we are interested in is actually the specific kind of biological interaction between certain molecules. Okay, so for example, everybody knows that enzyme reacts with its substrate in a very specific manner, or antigen reacts with the antibody, isn't it? Then what else is there? A ligand will interact with its receptor.
Excuse me, sir, the slide is not visible.
Okay, I'll share it again. Can you see that now? Is that okay?
Yes, sir.
Okay. So there is a specific interaction between antigens and antibodies, or ligands and the receptors. Okay, so that interaction is used in this kind of chromatography, where if you want to separate a particular molecule, then it's a specific complementary molecule, okay, which is going to interact with that molecule on account of the complementarity of shape. Okay, so that molecule is fixed on the affinity chromatography column. Okay, so here the stationary phase has a bound certain molecule, and that molecule is going to specifically interact with the sample component. Okay, only a particular sample component which we are interested in. And therefore nothing else will bind to that column. All the other proteins, they will be eluted from the column with the mobile phase. They will just go away from the column. And what will remain on the column is only the specific molecule, which is complementary to the group which is present on that chromatographic column. Is that clear? And therefore, this particular method is a kind of an absolute purification method. Absolute purification method means what? In one single step, your molecule of interest will bind to the column. All the other molecules will move away from the column. They will go out of the column. Okay. Your desired molecule only will stick to the column. Is that clear? Yes or no?
Yes, sir.
So initially this method was developed for separation of nucleotides or nucleic acids, immunoglobulins or antigens, membrane receptors. Okay, but now even for enzymes, it was used for enzymes. But nowadays, this particular technique is so versatile, or it is so useful, that almost practically anything in the world can be separated using affinity chromatography. Because we know that we can generate antibodies for anything, isn't it? Antibodies can be made for anything. And therefore, if you have the antibodies binding to the stationary phase, then the complementary molecules can be separated by affinity chromatography. This is a representation here. I have put capital M is the macromolecule, and L stands for any ligand, okay, which is complementary to the macromolecule. And then there is a kind of equilibrium. There is a complex formation between the macromolecule and the ligand. Okay. And therefore, if we put the complex mixture of the sample, which contains your specific or desired molecule, desired compound, then the desired compound will bind to the column. All everything else will be removed from the column. Okay, it will be washed away by the mobile phase. And then this desired complex, which is a component which is binding to the column, can be recovered later by what is called as displacement. Okay, so how do we carry out displacement? For example, if I want to purify enzyme, then what I can do is I can bind the pseudo substrate for that enzyme. Not the real substrate, but a substrate analog can be used. Okay, this substrate analog is in that stationary phase, right? Shall I show it on the whiteboard? Can you all understand what I'm saying? Otherwise, I'll make use of the whiteboard. Can you all see my whiteboard?
Yes, sir.
Yes, sir.
Okay. So what I have here is maybe this is a ligand, or this could also be a substrate for a particular enzyme. Okay, not the real substrate, because the real substrate will be converted into the product by enzyme substrate reaction. So what we have is a substrate analog, which looks like the substrate, but it doesn't react with the enzyme like a substrate. Okay, and then whatever sample component I'm adding here through this column, okay, I'm going to add the sample mixture, and in this sample mixture the desired molecule is present. Okay, so that desired molecule, maybe I'll show it with this color. Just a minute. Okay, so this is the desired molecule, which is now binding. Okay, and any other molecule which is not of that particular type, okay, which cannot interact with the ligand, that molecule is going to come out of the column. Is that clear? And the desired molecule will bind to the column. Is that clear to everybody? Okay. Yes or no?
Yes, sir.
Okay.
Yes, sir.
So now the next question is how to elute this. Whatever is sticking to the column, the desired molecule, how can we remove that from the column? And for that, we again have a very interesting strategy. Okay. So what we now do is we have this same molecule, this molecule, okay, the green colored molecule, which is already attached to the chromatographic medium. Okay. This attachment is by covalent interaction. I'll just try to write that here. Okay, this one actually is a covalent interaction. Let me change the color. This one. Okay, this interaction is a covalent interaction. Okay, so now what I'm going to do is I will have this same molecule, that is the ligand, which was attached to the stationary phase or the support material. Okay, but that is in a free state. Okay, let me draw it here. Free state. Can you all see that? Can you all see that?
Yes, sir.
Yes,
Yes,
sir.
sir.
Okay, although my drawing is not very accurate, but you can imagine that whatever green colored ligand was there on that column, the same ligand I'm taking in a free state, that is in a solution form, and that I'm adding to the mobile phase. Okay, this is added to the mobile phase. So now can you imagine what will happen? This is going to interact with this component, which is sticking. Okay, the desired molecule, which is already binding to the column. Okay, the pink colored ligand, okay, which is in the free state, will compete with this green colored ligand, which is on the column, which is bound to the column. So there is a competition now. And when I say competition, always remember that these molecules, the pink colored molecules, are in excess. I'm adding that in excess. Okay. And another thing, before I there is any confusion, you should understand that the interaction of this red colored molecule, that is the desired molecule, with the green colored ligand, which is bound to the column, this interaction is not a permanent interaction. Just remember that. The red colored molecule will bind to the green colored molecule and it will come off again after some time. Okay, and then it will bind to another green colored molecule. Just a minute. Here there is one more. Okay, so red colored molecule can come out of this place here, position number 1. Let me also put that here. Position number 1. Okay, here it is position number 2. This probably is position number 3. Okay, so this molecule from position number 1 can get removed from there and it can bind to position number 3. Molecule from position number 2 can go to position number 3 or 4 or 5. Okay, so this is an equilibrium reaction where the molecules are not permanently bound to the ligand, but they can come off and they can again bind to next molecule. Okay, so these interactions are happening very dynamically. Is that clear to you? Whereas any other molecule which is in the sample, which is not specific, will not be able to interact with any of these green colored ligands. And therefore they will come out of the column very rapidly. Okay, now what happens is when I add a pink colored ligand, which is again a competitor to the green colored ligand, and that is also in excess. And because if it is in excess, the moment the molecule from position number 1 comes off because of the equilibrium, okay, it will come off from that green colored molecule, and immediately there are surrounding maybe 10 or 15 pink colored ligands, and therefore that red colored molecule, instead of binding to the green colored molecule on position number 1, it will bind to the pink colored ligand or pink colored molecule. Is that clear? सगळ्यांना कळतंय का?
Yes, sir.
Oh, sir.
And then if the red color molecule now binds to the pink colored molecule, let me show that. Oh, sorry. Okay, so interaction is now happening like this. Red color molecule from position number 1 has come off and it is binding to the pink colored molecule. Because the pink colored molecule are in excess, there is a great competition offered by them. And now this particular complex, okay, this is a complex now. And that complex is now going to come out of the column because this red color molecule now has no opportunity to bind to the column. Okay, the complex will come out. It will be eluted from the column. It will come out from the outlet of the column. Is that clear? And as we are taking the fractions, okay, probably some fractions, okay, successive fractions, maybe fraction number 13, 14, 15. Okay, these 3 fractions will contain this complex now. Okay, free ligand bound to the desired molecule. That complex is there. Okay, of course, remember that this interaction with the ligand is never a permanent interaction. The molecules will dissociate, they will reassociate, dissociate, again they will reassociate. This is happening continuously. Is that okay?
Yes, sir.
Okay. Now my question to you is, now we have this complex with us, okay? When we pool together the fractions, in one of the pools we have this complex. Okay. The next question to you is again, how to separate the complex? Because we want only the desired molecule. We don't want that ligand associated with it for any of our application further, okay, or any of our analysis maybe. We don't want the complex to be there. We want only the purified component. So how are we going to purify that? Okay, so that was about elution. If you can see my slide again, I have already told you in the diagrammatic way how we can separate the desired protein from the column. Okay, and then my question to you again is how to separate the complex. And for that, once again, I have the same answer. Okay, what is that same answer?
Dialysis.
Yes. कोणी म्हटलं हे? कोणी सांग?
So Vaidehi.
Vaidehi, okay. Yes, Vaidehi is right. Okay. So once again, we have the same answer. We can carry out dialysis. Okay, dialysis कसं करणार आपण? How can we carry out dialysis? मी हे सगळं इरेज करतो पहिल्यांदा. I hope you have understood this diagram. And then I'll go for another diagram. Okay, for example, I have this beaker. Okay, and in this beaker, I have distilled water. Okay, and inside the distilled water, I am putting this dialysis bag in the form of a chocolate. So chocolate सारखं दिसतं ना हे मस्त. And inside this dialysis bag, I have this sample. Okay, my sample is inside. Is that clear? आता ह्याच्यामध्ये काय आहे सॅम्पल मध्ये? I'll just try to put one complex inside. Okay, this is the ligand, and this is my molecule of interest. It may be a receptor molecule or any kind of a protein. Okay, and then when I carry out dialysis, this is distilled water. Okay, let me put that also here. Distilled water. Okay, so because I told you that there is a continuous association and dissociation of these molecules. They are not permanently bound, and therefore you can just imagine that whenever there is a dissociation of these molecules, okay, this molecule, you can imagine that there is a high concentration of these ligand molecules, free ligands, because of the association dissociation process. And the moment the opportunity is there, this molecule, okay, let me, okay, again I'll show it diagrammatically. Some of the molecules are free like this. Okay, some of them are free because of the association dissociation process. Is that clear to everybody? And the moment these molecules, there's a certain number of free ligands, okay, because it is a dialysis tubing, these ligands are going to come out of this dialysis tube. Is that clear to everybody? And I'm going to change this distilled water repeatedly. Okay, 5 times, 6 times, or whatever, 3 times, depending upon the level of purity that you get. Okay, so when you do the same process 3 times or 5 times, then most of these ligands are going to come out of the dialysis bag. And what will remain inside the dialysis bag is the desired molecule. Why will this ligand come out? Because the ligand is a small molecular weight molecule. It is a very small molecule which can come out of the dialysis tube. Whereas the protein, which we have separated, that protein is not a very small molecule. It is a macromolecule, and therefore it will not come out of the dialysis bag. I hope everybody has understood this. Akash?
Yeah, yes, sir.
Have you understood?
Yeah, yes, clear, sir.
Okay. Then there is something that you should know about how should be the chromatographic medium. Okay, the ideal, if you want to know about the ideal chromatographic matrix or chromatographic medium, then it should have sufficient chemical groups to which the ligand can be covalently coupled. So more the number of ligands are there on that stationary phase or the supporting material, more will be the interaction with the desired molecule. Okay, so the chemical group should be there to which you can attach the ligand. Then another thing is the chemical groups must not only be there, but they should also be stable under whatever conditions we are employing. Okay, and then because of the continuous binding and dissociation, the chemical groups must remain stable, isn't it? There should be no damage to these chemical groups, or they should not get removed from the from the support material. Otherwise, the efficiency of the column will drop down. Okay, Then another thing is the chemical groups should not interact with any other molecule which may be there in your sample mixture. Otherwise, the purpose will be defeated. Instead of getting your desired molecule attached on the column, you can get anything else which will bind to the column by non-specific interaction. So that should not happen. Then of course, the mobile phase should be able to flow properly, okay, under whatever conditions you are employing. If it is HPLC, then we are going to apply pressure also. And therefore, under that pressure, the matrix material should be stable. The chemical groups also should be stable, okay, and they should allow the proper flow of the mobile phase, right? So different types of the chromatographic media are used. For example, agarose beads, dextran, polyacrylamide, polymethacrylate, polystyrene, cellulose, silica, glass, porous glass. So all these different materials, organic as well as inorganic materials, are used to make such affinity columns. Okay, then coming to the ligands which can be attached to the support material. Either we have those ligands which can exactly bind only to the desired molecule. So when that is the case, we call it as absolute specificity, or sometimes what is there is a group of different molecules can bind to the same column. For example, if you bind 5-AMP to the column, then this 5-AMP can bind to many different enzymes which belong to this particular class, that is